My name is Peter Andras. I am a professor at Edinburgh Napier University. This is my lecture series on evolution of complex systems. This is lecture 4 about cells and the genome. This is part 1 of lecture 4. So in this lecture, we will look at cells and proteins, how these form systems. So first, we will consider the cell systems as described traditionally. Then we look at the language of the cell and memories, in particular in the context of the cell, which we will see that we interpret the RNA and DNA molecules as such memories. So let's start with cells. You will find cells everywhere in all living organisms, plants, animals, bacteria, archaea. Cells are everywhere. These form tissues in organisms, or they live as single-cell organisms. You will find many examples, so for example, neuron, muscle cells, skin cells, and so on. There are many kinds of cells in organisms. What you see on the screen on the left side are bacteria, while on the right side is an image of a neuron. And generally what you can find is that bacteria are generally smaller than the cells in multicellular organisms. Now, if you look at the cell with a microscope, then you will find in most of the cells many components. Now, what you see on the screen is a cell from eukaryotic organism, which means that they have a nucleus. These cells have more structures, so we will consider these for this purpose. So the cell has a cell membrane. Sometimes it has a cell wall on the top of the cell membrane. Inside of the cell, in these eukaryotic cells, there is the nucleus that you see the kind of large brown spot in the middle. Inside of it is the nucleolus, which is a kind of phase-separated part of the nucleus with particular function. And the nucleus is enveloped in a nuclear envelope, which connects to the internal membrane structure in the cell, which is the endoplasmic reticulum. And then you have the centrosome, where mitochondria, various vesicles and membranes, and all kinds of structural fibers like microtubules, actin fibers, and so on. So one way to describe the cell is that you look at what you see in the cell and you describe the various components as they appear and their function. Now, if you look at the cell, what you see is that it's delimited from the exterior. It has a cell membrane, and sometimes it has a cell wall on the top of the cell membrane. This is a typical semi-permeable structure made of a lipid membrane with proteins inserted in it, which work as ion channels or signaling molecules and so on, or receptors. Now, inside of the cell membrane is the cytoplasm, which also contains the various cellular organelles. The cytoplasm is kind of watery solution, it's kind of gel-like solution of proteins and various other molecules with some membranes and possibly other structural components in them. The cellular organelles are typically enveloped in their own membrane, or they are formed of a certain structured membrane, and they perform various structures, various functions. Some of them secrete molecules, some of them digest molecules or nutrients in general. Some parts of the cell, their centromere and connected fibers are involved in the cellular motion, and so on. So in general, what you see is that in cytoplasm, there are the many molecules that come in and leave the cells and mix together, move around, and there are these various organelles which perform various functions within the cell. Now, if you look inside of the nucleus, you will find DNA, the chromatin, this kind of protein-based structure which keeps the DNA packaged, and RNA molecules, and of course you find also many proteins around them. Now, this is a case where the cells have a nucleus, like the eukaryotic cells. In bacteria and archaea, the DNA is the spiral structure that is distributed across the cell. It is not packaged in a nucleus. Now, in general, the DNA contains the code that describes the components of the cell in terms of peptides which will form proteins. So these, the genes, are the parts of the DNA which encode the proteins. The DNA has 4 letters, if you like, the adenine, cytosine, guanine, and thymine, ACGT. Now, these nucleobases are the only 4 nucleobases that are used in the composition of the DNA. These are connected to a sugar molecule, and phosphorylated sugar backbone holds these nucleobases ordered. They all form pairs, and they encode proteins and other, they also contribute in many other ways to the regulation of how proteins are produced. So in general, the DNA is organized in genes and non-coding segments. And non-coding segments have mainly regulatory roles. They regulate when the coding segments can be decoded, when they cannot be, and to what extent, and so on. And as the DNA gets decoded, it is transcribed into an RNA, and then the RNA is transported outside of the nucleus after a maturation process. And then at the ribosomes, which are little organelles on the endoplasmic reticulum, the RNAs drive the process that generates the proteins. Now, if you look at the behavior of the cell, what you see is that there are a range of behaviors. So they bring in molecules from outside, they bring in larger resource particles, for example, another cell or a fragment of another cell. They digest the things that are brought in from the outside of the cell. They secrete the waste products which are released into the outside of the cell. They generate all kinds of proteins which become the building blocks of the cell, of the various membranes, organelles, cell membrane, and so on. A major behavior of the cells is the self-replication or the multiplication. When the cell grows and then divides, generating 2 cells of the same kind. And finally, the self-defense, which is mainly implemented in terms of secretion of self-defensive molecules, proteins, but also through the movement of the movement behavior of the cell. So far, we looked at the cell in the traditional way by describing the various components, the functionality of these, and the behaviors of the cells. Now, we can look at the cell behavior in a different way. So, as I said, the DNA's segments, the genes, are copied through transcription into RNAs. The RNAs drive the production of proteins at the ribosomes. Now, these proteins, these long chains of amino acids, they really participate in the driving of the cell behavior in all respects. So proteins interact with other molecules, lipids, sugars, phosphates, and so on. Through these interactions with other molecules, they generate further other molecules in the cell, and the patterns of these interactions deliver the behavior of the cell. Now, if you look at how proteins work, some work on selecting resources, like what kind of ion should be let in the channel proteins. Others deal with self-defense as receptor proteins, sensing whether there is something that requires defense responses. Others catalyze reactions between other proteins to generate further more proteins, and so on. So what you see here is that compared to the traditional description, which looks more at the phenomena and finds features of them and describes them, this alternative view of describing cell behavior focuses on how molecules participate, in particular how proteins participate in these processes. What is their role, and how do they deliver their role through interacting with various other molecules? So the proteins are formed by amino acids. These are such chains of amino acids, and in most of the living cells, the proteins are formed by 20 amino acids. There are many more amino acids, but only 20 of these amino acids are used in the formation of the proteins in living cells. Here on the screen you see these 20 amino acids. I know that there are two further amino acids, the pyrolysin and the cysteine, which are also used relatively rarely in proteins in living cells, but in most of the cases, these 20 amino acids are used to compose the proteins of living cells. Now, it is not totally clear why exactly these 20 amino acids are used out of the hundreds of amino acids that could be used, but this is how it is. Cells use only these restricted set of amino acids to form the proteins inside of the cells. Now, if you look at these protein molecules, what you find is I have very long sequences of amino acids which coil up and form kind of dense structure which has sheets and helices. The helices have shorter periods, while the sheets have longer chunks in the coiled up structure. Now, the primary structure is the sequence of the amino acids. The secondary structure is formed by these alpha helices and the beta sheets, which compose the proteins. Now, the tertiary structure is how these alpha helices and beta sheets fold onto each other to form the protein. Now, what drives the formation of the helices and sheets and the folding together of the protein are the interactions between parts of the amino acids which form the proteins. The amino acids have a distribution of electrons within the molecule, which is such that some parts of these amino acid components become positively charged and some may become negatively charged, depending on the amino acid and on the context. And while others don't get charged because their distribution of electrons is different. And as a result of these charged and also non-charged nature of amino acids, the forces that pull together these components of the molecules drives the formation of the alpha helices and beta sheets, and then furthermore, they drive the formation of the protein folding. There are various bonds that form weak and not so weak bonds that form between various parts of these molecules. And these bonds hold together this folded structure of the proteins. Now, the quaternary structure of the proteins is how the active sites of the proteins emerge and change. Now, quite often, the proteins act together. Quite often you have multiple copies of the same protein forming a larger structure, or a few, maybe a couple of proteins, but again multiple, a couple of copies of them forming a new structure. So these can be pores or ion channels or various receptor structures and so on in the cell. Now, the quaternary structure, the active sites, is what determines how the protein is actually working. So these active sites are on the surface of the proteins, which are active in the sense that they can interact with other molecules. Typically, these parts have electron distribution which creates some charged structure, with some parts being positive, some parts being negative, or in other ways may facilitate interaction with other molecules. Now, these active sites on the surface of the protein, typically it is seen as a ball, or occasionally it's a thread. On the surface of this molecule, these active sites change depending on how it interacts with other molecules. So, for example, the proteins may link with sugar molecules and other proteins to produce energy storage molecules. And there are many other molecular interactions. Ultimately, all behaviors within the cells are delivered through these interactions of proteins. So this gives you another view of how to see the cells and cellular behaviors as driven by interactions of proteins that form these potentially quite large intermediate complexes and deliver their interactions. What you see on the screen is one particular functional pathway in cells, which includes a number of enzymes, enzyme proteins, which catalyze a number of reactions involving other proteins and possibly other molecules, and delivers these functional cycles. So an alternative way of looking at the cells is that you see these functional cycles, how they form together the delivery of the behaviors of the cell through the generation of right kinds of proteins at the right time, right place that interact with other molecules in the right ways. And signaling pathways are pathways where proteins which act as receptors get interacted by other molecules outside of the cell. Possibly these can be other proteins, but possibly other smaller molecules. And this interaction changes the conformation of these receptor molecules, which means that their part that is inside of the cell changes the conformation, changes active sites on it, so it can interact with other molecules and triggers a cascade of molecular interactions. This is a signaling pathway. So, for example, a neurotransmitter molecule may attach to a receptor, which triggers the opening of ion channels through a number of protein interactions and also interactions involving other molecules and ions, which lead to a behavior of the cell. Quite often, signaling pathways also lead to activation of certain parts in the nucleus, impacting what kind of proteins are produced within the cell. So, What I discussed so far is that on one side you can describe cells in the traditional way, looking at what's there, what kind of phenomena can you see, and how can you describe it. The alternative way that I offered is that you can describe the cell as a system of molecular interactions where proteins play the key role, and you can describe all behaviors of the cells in these terms, in terms of these kind of interactions or patterns of these interactions. So, taking the view of the systems theory that we introduced in the previous lectures, we can look at the cell as a communication system. So here, the spatial-temporal patterns of protein interactions will be seen as communications. So the communication units are the molecules, primarily the proteins, and the communications are the interactions between these molecules and the patterns of these interactions. So we discussed the signaling pathways, how they involve a long sequence of interactions between specific molecules, which trigger further interactions in the cell. Or we can look at any other aspect of the cell behavior, for example, cell wall synthesis, where the cell senses that cell wall molecules needs to be synthesized. In response to that trigger, the right molecular interactions follow, which generate the components of the cell walls. And these molecules are assembled again through a series of molecular interactions. They get transported to the membrane of the cell. They get transported through it and added into the cell wall to grow the cell wall, for example, when the cell grows. So, really, all behaviors of the cells can be seen implemented through these protein interaction patterns and sequences of these protein interactions. So, how referencing might work? Well, given that the molecular interactions depend on the availability of molecules that are required for the interactions, and all these other molecules, particularly proteins, which are the major players in these interactions, are the products of other molecular interactions, we can see the involvement of these proteins into these interactions as a reference to the interactions which generated these proteins. So, for example, in the functional cycles, the example that I showed you earlier, you have many enzymes participating at all of these enzyme-supported steps. A new protein or several new proteins are generated. Typically, it's one or a protein is modified quite often. And each of these steps leads to a product which participates in another molecular interaction, possibly again acting as an enzyme catalyst of other interactions, leading to production of further molecules. And through these patterns of molecular interactions, the cell behaviors are delivered. Now, what you see in the cells is that there are many, many molecular interactions inside of the cell. As I said, it is a gel-like watery mixture that is quite densely packed with molecules which interact very frequently. Compared to these interactions outside of the cell, there are again many molecular interactions, but much fewer in terms of frequency or density. How often these interactions happen? It happens much fewer. Like this, the inside of the cell is quite densely packed, so there are many interactions that can take place. Outside of the cell is much, much less densely packed, so the likelihood of interactions is much smaller. As I said, there are still many interactions, but much less than inside of the cell. So what you see again is that there is this boundary of rare communications and very frequent communications inside. What's inside? That is the communication system. where the communications are these protein interactions and other molecular interactions. And this is how we can see the cell as a system of molecular interactions. And the boundary, in this case, there is a physical boundary that we can identify, which is the cell membrane and possibly the cell wall in addition to that. Now, the actions of the cell, these can be seen as sequences or patterns of sequences of communications. So here the communications are the molecular interactions, and these actions of the cell, which are impacts on the environment, are, for example, the production of proteins that get incorporated into the cell membrane as cells, as receptors or signaling molecules. These patterns may lead to the production of secretions, which are molecules released into the environment of the cell, or they may be involved in the production of the movement of the cell. So, for example, bacteria may move by undulating their flagellum, or some cells may move by moving of small appendages, or they may move like an amoeba, and so on. There are many ways how cells can move, and all these movement behaviors, which are effectively impacting the environment, at least mechanically, are produced by a sequence of molecular interactions involving many proteins. Now, these actions are referenced by other communications within the cell because those molecular interactions which deliver the cell's action on the environment, they also generate molecules that interact inside of the cell. So in this way, the molecular interactions, the communications within the cell, reference the actions of the cell. Now, let's look at the perceptions of the cell. In this case, you may have receptor molecules which are in the cell membrane, and these interact or bind to receptor messenger molecules. So the receptors and messenger molecules bind together. In response, the receptor molecule changes its conformation and interacts with other molecules inside of the cell, which otherwise it wouldn't interact with. Now, as a result of these interactions, other changes may happen. For example, pores or channels may open on the cell membrane, and ions and possibly other molecules may enter the cell. And then again, this will lead to further molecular interactions, either because the ionic balance changes or because molecules get into the cell that directly interact with other molecules. So, in general, what you see is that, again, the perceptions of the cell are implemented through molecular interactions involving proteins. And again, these produce further proteins which interact with other molecules, other proteins within the cell. So in this way, these perceptions of the cell get referenced by communications within the cell. So, effectively, we already put it together that the cell can be seen as a system of protein interactions, where the protein interactions are seen as the communications. And these communications are essentially about defining what is the cell and what is not the cell. What's the cell's identity? Because essentially these communications are about defining what is useful for the cell, which are the resource molecules that are useful and can be used to generate further proteins that compose the cell and maintain the molecular interactions in the cell, and which are the ones that should not be incorporated, which should be excluded from the cell. So, in an abstract way, what you can say is that these molecular interactions in the cell are really about deciding how to maintain the cell, which molecular interactions are the right kind of molecular interactions, and which are the ones that are not. So the aim of this cell as a communication system is to reproduce and expand itself, which is indeed what cells do if you look at them in a functional phenomenological way as well. So now we came to a point that we define the cells as systems of communications, where the communications are molecular interactions and primary protein interactions. And we said that the aim of this cell is to reproduce, and all these interactions are about defining what's a cell, what's not, what is good for the cell, what's not good for the cell. So this of course leads to the concept of the meaningful and meaningless communications within the cell. And you can see that in this context, the meaningful communications are those patterns and sequences of protein interactions that lead to the incorporation or elimination into the cell or from the cell, elimination from the cell of metabolites resulting new protein interactions. These protein interactions may not be directly related to the incorporation and elimination of other molecules, but indirectly, as referenced molecular interactions, they are related to. So all those molecular interactions that lead to further molecular interactions within the cell to maintain the cell itself, these are meaningful communications. The meaningless communications on the other side are the molecular interactions that lead to products that cannot interact with other molecules, cannot be part of the cell, and possibly they are not even eliminated from the cell, so they get accumulated and damage the cell functionality. If you like, they constitute identity violations within the cell because they don't fit the identity of the cell, they don't fit the molecular interaction expectations within the cell. To give you some examples, for example, you may consider the antibiotics, toxins, or prions. The antibiotics are molecules which are used against bacteria. Typically, what they do, they interact with certain molecules. For example, penicillin interacts with a small set of proteins which are called the penicillin binding proteins. As it happens, these proteins have very key functionality in the cells. They drive or contribute in a key way to the synthesis of the cell wall. If you add penicillin or other antibiotics to these bacteria, to the environment of them, the penicillin binds to these penicillin binding proteins. If the bacterium is not able to eliminate the penicillin, it blocks the cell wall synthesis in the bacterium, and as the bacterium grows and would need to synthesize more cell wall to allow its growth, effectively blows up and the bacterium dies. And how this works is that the penicillin interacts with these few molecules, changes the conformation of these molecules, blocking their normal interactions with other molecules, so the cell wall synthesis stops. Toxins quite often work in the same way, or at least in a similar way, in the sense that they bind to molecules, interfering with their normal functionality and preventing the delivery of this normal functionality, so the behavior of cells is damaged and potentially the cells get killed. Prions are another case of strange molecules. So these are protein molecules which have a certain conformation which is such that it triggers the copying of this conformation by other proteins of the same kind. What is unfortunate with some of these prions is that when they take this conformation, they become unable to deliver their other molecular interaction rules. So again, the molecular interaction system of the cell gets damaged because the expected interactions don't happen, and ultimately the cells get damaged. And they become less functional or potentially may die. Now, these molecules are antibiotics, toxins, prions, cause faulty communications within the cell and potentially cause error situations in the cell. If these happen very often, that leads to the failure of the cell system, which in practice might just mean the death of the cell because of the dysfunctionality, because it cannot grow or because it cannot release the base products or some other key functionality of the cell breaks down and then the cell dies. Now, what we say here is that we can see the cells as systems of protein interactions and molecular interactions, and in this context, the language of the cell is the pattern or sequence of these molecular interactions. The grammar of the cell are these rules which tell you how these molecular interactions follow or can compose together. The referencing rules tell you what molecules need to be produced and what molecules need to be in what state to certain molecular interactions to happen. While on the other side, the continuation rules tell you that given the presence of a set of molecules at the right place simultaneously, what kind of interactions may happen and what kind of new molecular interaction can take place, which generate possibly new molecules. Now, what you need to see here is that quite often the proteins which act as catalysts get into an activated state where they can help energetically to other molecular interactions to happen which otherwise couldn't happen. And as a result of this, some of these molecules change. The enzyme molecule quite often returns to its pre-activated state, while some of the other molecules may break up or may join together or may undergo some kind of modification, for example, adding of a methyl group to it or adding of a phosphate group to it, and so on. So, all these referencing and continuation rules together form the grammar of the language of the cell. Now, I have to say that we can talk about the language of the cell in this sense. We don't know yet in actual details of how does it work. There is a wide range of knowledge about what kind of molecular interactions happen, in particular what kind of protein interaction happens in the cell. There are new techniques which can be used to find out in a more precise way of when and where these certain molecular interactions happen. However, our current knowledge is still limited about what is actually ongoing in the cell, although we know quite a lot and the knowledge is increasing. For this reason, while in principle we can talk about the language of the cell as these referencing and continuation rules that drive the protein interactions inside of cells, this is so far just in principle because we don't really know the fine details of the actual rules of how cells work. However, this principled view already helps in understanding and viewing the cells in a different way and understanding how they deliver their functionality as a communication system. Now, structures, as we said, these are constraints on communications, constraints on referencing and continuation rules. In the context of the cells, these structures are particular constraints that apply to protein interactions, which constrain them and make them either more or less likely to happen. And these constraints can be, for example, that certain proteins have to be inside of a vesicle, where potentially the pH is regulated up or down depending on what is needed to facilitate certain molecular interactions. Or it might have to be inside of a membrane, or it might need to be bound to some other molecule in order to deliver certain molecular interactions. So, for example, proteins may be marked by adding to them certain other molecular components. So, for example, ubiquitination, it is a way of marking proteins for destruction. There are other ways which mark proteins for transport, for example, to inside of certain vesicles or across the cell membrane and so on. So in general, what you see is that there are constraints implemented in terms of location or binding of proteins in certain ways, which constrain their interactions, and constraining their interactions effectively leads them to become parts of certain structures. So these structured or constrained molecular interactions effectively gather together and get grouped together in the context of various components of the cell, be them certain kind of membranes, a vesicle, or in general cellular organelles. Quite often, there is a particular functionality associated with these particular molecular interaction patterns. So, for example, you may look at the mitochondria, which deals with the cellular respiration, which is the use of oxygen to generate energy inside of the mitochondria. So again, it is certain kind of molecular interactions which happen only in the mitochondria, which are constrained and deliver a particular functionality. Or if you look at the centrosome, again, it's a particular molecular structure with particular molecular interactions, which is at the core of movement of the cell. Or in the same way, you may look at the cellular digestion in the various vacuoles and lysosomes and other vesicles, which create particular conditions, for example, high pH, high proton concentration, which facilitates certain molecular interactions. Now, we noted about the memories, that memories are communications which facilitate the referencing of past communications or the reproduction of these past molecular communications. So in this case, we are talking about molecular interactions. So let's look at what facilitates molecular interactions, or if you like, the remembering of molecular interactions. And here what we can point out is that the mRNA molecules inside of the cell are the ones which facilitate certain kinds of molecular interactions involving proteins. And these are the interactions where one amino acid is added to a chain of amino acids. Effectively, this is the protein synthesis process. So if you like, the mRNAs facilitate this set of molecular interactions, which ultimately leads to the production of the proteins inside of the cell. Now, it's also important to note that there is a variation. So, for example, it can be that some mRNAs in some cells, in some species, may encode a combination of molecules that are encoded separately in other species. So these molecules can represent, if you like, interactions of proteins as well. For perhaps in an abstract way, but effectively what this suggests is that if those molecular interactions which require these corresponding proteins to act together are important, then mRNAs may represent these interactions by encoding a by jointly encoding the combination of these proteins as well. Now, the RNAs, this way, mRNAs in particular, they generate the right kind of proteins which deliver the right kind of behavior of the cells. So what we think here is that you can see the mRNAs as memories which facilitate the reproduction of these molecular interactions, facilitating the reproduction of the interactions between chains of amino acids and single amino acids, which involves the mRNAs and the tRNAs, and also the rRNAs, which are in the ribosome. And through the facilitation of the production of these proteins, it facilitates the production of all other protein interactions within the cells. And there is also one interesting note that around the mid-2000s there was an interesting experiment where they showed that in the Arabidopsis plant, after the plant was selected to have some certain deformities or developmental changes, it returned to the normal development pattern, although they analyzed and they showed that this was not present in the DNA. The DNA encoded the altered development pattern. Now, what has been suggested is that this is actually mediated by RNA communications by representing the correct proteins in RNAs which are retained in the gametes. And through this way, the normal functionality of these plant cells or the plant organisms is restored. Now, there are many other RNA molecules in the cell. So as I mentioned, there are the mRNAs, these are the messenger RNAs, which encode the proteins. There are the tRNAs, which are transfer RNAs, which pick up amino acids and deliver them to the ribosomes where they get attached to the sequence of amino acids. There are the rRNAs, the ribosomal RNAs, which are part of the ribosomes, and they facilitate all these interactions with the mRNAs, tRNAs, and the sequence of amino acids. And a number of other non-coding RNAs, small interfering RNA, the siRNA, microRNAs, circular RNAs, and many other RNAs. Many of these non-coding RNA molecules interact with premature mRNA molecules inside of the nucleus before the finally formed mRNA molecule is released from the nucleus. Through these interactions between the RNAs, there is a splicing process where the premature mRNA molecules may be cut in parts and rejoined. And through these processes, the original transcripts from the DNA get transformed into the mature mRNA, which actually encodes the proteins that are produced in the cytoplasm. This process can lead to multiple variants of the mature mRNAs generated from the same mRNA, premature mRNA, depending on how these RNA interactions work out. There are also other small RNAs which may interact with other RNAs and possibly proteins as well, which mark them for destruction or mark them for silencing and so on. So it is a quite complex process of interactions between RNA molecules. What we said is that the RNA molecules can be seen as the memories of protein interactions, directly memories of interactions between single amino acid and the chain of amino acid, but also memories in more general sense of proteins and interacting proteins that are required for the functionality of the cell. Now, the interactions between the RNA molecules can be seen as the information communications within the cell, and as such, the RNA interactions are seen as the primary information subsystem of the cell. Just to note here is that there is the concept of the RNA world, which suggests that originally there was RNA, and these RNA molecules interacted and generated proteins, and that's how life might have started. And clearly, what we said so far somewhat is reasonably aligned with this approach, where we say that RNA molecules and interactions of them can form a primary information system of a communication system, where the communication system is the system of molecular interactions involving proteins. And this is the core information system, which is required to generate and manage the memories, manage the information about the system. Now, as I said, the RNA molecules are produced by copying segments of the DNA molecules. The difference between them is that in the RNA, the sugar chain is different. It's a ribose chain, while in the DNA, the sugar chain is a deoxyribose based sugar chain. And there's also another difference, that instead of the thymine, the T letter in the DNA, the RNA contains the U letter, the uracil base, which is different from the thymine base that is used in the DNA. So in the now, this copying works, so the T's are copied as U's and the rest is copied as it is in the DNA. This is a transcription process. So first, the premature mRNA is generated, which contains several RNAs which interact and generate in the end the mature RNA. Now, what you can see here is that the DNA encodes all these components of the premature mRNA, which get cut around, spliced up, and interact together to generate the final mature RNA. So in a sense, the DNA encodes the interactions between these RNA molecules as well, which lead to the generation of the mature mRNAs, which are later used to translate them into proteins. So in this way, you can see the DNA as memories of the RNA interactions. So the DNA, in that sense, constitutes a further layer of information processing within the cell, which is the memories of the primary information processing system, which is implemented by RNAs and RNA interactions. So, to summarize, we looked at communications as protein interactions. We said that you can see the cell through the system of protein interactions, and you can see all behaviors of cell delivered in terms of protein interactions. We noted that, of course, there is the traditional view of the cells, but actually all elements of that can be replicated by a corresponding protein interaction based approach of the description of what happens in the cell. Then we define the cell as a system of such communications, where the communications are the protein interactions, and we define the cell's language, the continuation and referencing rules of protein interactions, which tell you what other interaction must have happened before to generate a third particle interaction, and also tells you that given a set of molecular interactions that happen with involving protein, what further molecular interactions can happen given the products of these previous interactions. And we said that this constitutes the cell language. We also noted that at the moment this can be described in principle, and although we know a lot about what kind of molecular interactions happen in real cells, actually we are still limited in the understanding of what kind of molecular interactions happen at what time and where within cells. Now, the actions and perceptions of cells are described in terms of protein interactions, where the actions are the protein interactions that lead to some impact on the environment, like movement of the cell or secretion of some molecule, and the perceptions are the molecular interactions which are represent the impacts of the environment, for example, triggering of receptor molecules, or mechanical impact on the cell, or heat impact on the cell, which all trigger certain molecular interactions, and those molecular interactions represent the perceptions of the cell. Then we discussed about what is meaningful and meaningless. We said that all those molecular interactions which lead to further continuations of molecular interactions and the maintenance of the cell, these are meaningful, and the ones which don't do this are the meaningless. For example, toxins, antibiotics, prions are molecules which generate molecular interactions that are meaningless in this sense. Then we noted the role of structures and subsystems. So the structures are constraints, and in the context of cells, these may mean being embedded in a certain place, or being marked or bound to a certain other molecule. And he said that these structures may constitute subsystems within the cell, which are functional subsystems such as respiration, the mitochondria, or the centriole, which drives the movements of the cell. And finally, we discussed about memories. As I said, memories are communications which allow the reproduction of other communications. In this case, what we said is that RNAs constitute the primary memories and primary information subsystem of the cell, because they allow encoding of the molecular interactions which lead to the formation of the proteins, and ultimately in this way they encode all prerequisites of protein interactions within the cell. And we also noted that the DNA segments, which are transcribed primarily as primary transcripts, these are premature mRNA molecules which include a number of RNAs which interact, and through these interactions the actual mature products are generated. And in this way, we said that the DNA segments actually encode all the interactions between the RNAs, which are the memory processing interactions or the information communications within the cell, or within the primary information system of the cell. All right, so let's have a look at a few questions. Is it true that nucleic acids are part of proteins? Well, the answer is no. Nucleic acids are part of the DNA and RNA. These are the nucleic acids. These are not part of the proteins. Proteins are made of amino acids. Is it true that neurotransmitter molecules may become part of the cell system communications in neurons? Well, the molecules itself, as I said, these are the communication units. The communications themselves are the interactions between the molecules. So in that sense, the neurotransmitter molecules may participate in molecular interactions that are referenced within the cell and are part of the cell system. However, the molecules themselves are communication units, so they are not part of the communications. Next, is it true that the secretion products of a cell are part of the cell system? Well, again, the products themselves are molecules. Their interactions may be part of the cell system. Certainly, they reference molecular interactions that happen in the cell. However, most of these molecular interactions are likely to happen outside of the cell, and unless they lead to molecular interactions further in the cell, they may not be any longer generating molecular interactions for the cell system. However, quite often these secretions are secreted in order to generate the right environment for uptake of nutrients from the environment. So in that sense, potentially it is possible that the molecular interactions of the secretion products are further referenced by molecular interactions within the cell later. Is it true that a prion may cause meaningless communications within the cell? And the answer is yes, this is what we discussed. The prion distorts the conformation, the normal conformation of the proteins, and by doing this, prevents these molecules from delivering their normal role, so they lead to meaningless communications in the cell. Next question, is it true that the change of active sites of protein represents the effect of communication reference in the cell system? So, actually, if you look at it, so you have the protein which participates in molecular interactions, and as a result of that molecular interactions, the active sites change. So we are looking at an enzyme which is activated and then is active, facilitating molecular interaction, and then becomes inactive until it gets activated again. Now, in this way, the change of the active sites, yes, it does. You can see it as representing the effect of communication reference in the cell, because there is a communication and molecular interaction happening that is referencing a previous molecular interactions which delivered the enzyme, and the change on the in terms of active sites represents this referencing. Next, is it true that the penicillin may become part of a cell system if it contains penicillinase enzymes? Now, we haven't discussed about penicillinase. These are the penicillinase enzymes are proteins inside of bacteria which can decompose the penicillin molecules. And effectively, those bacteria which have the penicillinase enzyme are able to metabolize penicillin, and they are able to use the components of the penicillin molecule to produce useful molecules within the cell. So in this way, the penicillin, or rather the interactions of the penicillin molecule with other molecules within the cell system, they do become part of the cell system of molecular interactions within a cell, because when they interact with the penicillinase, they effectively become a resource molecule within the cell. Finally, can we see the ion combination rules as part of the cell language grammar? And the answer is no, because these ion combination rules, like sodium and chloride ions, how they combine, or protons and hydroxyl ions, these happen everywhere, not just in cells. They happen in the same way everywhere, so they are not part of the particular language or the grammar of the cell. Thank you.